So in this lesson, I'm going to talk a bit more about the cell cycle and mitosis. Before I can talk about the specifics of how the process occurs, we should talk about why cells divide in the first place. The first reason would be for reproduction. Asexually reproducing organisms must split in half in order to create more organisms like themselves. So examples of this would be like an amoeba, which is a protist. It's a single-celled eukaryotic organism, and also bacteria as well. They undergo a process called binary fission, which allows them to split in half. Another reason that cells divide is to help an organism grow in size. You start out as a zygote, then an embryo, then a fetus, then a baby, then an adult. In order to grow in size, you have to actually add more cells to your body in order to increase your size. And the last major reason anyway, that cells divide is for renewal and repair. For example, if you get cut or on your arm or on your finger, you actually lose cells where that cut occurred. So your body is going to send out a signal to the cells in the surrounding area that they need to start to divide and heal that cut. Also, you lose skin cells all the time. Whenever you hit something or touch something, you lose your skin cells, so they must constantly be replaced. Another reason is for regeneration. Some of your organs, the main organ that can regenerate is called the liver. The liver can actually grow back a large portion of itself using the process of mitosis. So the cell cycle is going to occur in three main stages. You can see two of the three main stages in the cell cycle diagram in the top right. Interphase includes G1, S, and G2, and then after interphase, it goes into the process of mitosis where the nucleus actually splits. A cell that's formed from a mitotic division has two kind of pathways that it can follow. It can follow the pathway that's shown on this screen to the left, where a cell grows and matures and then divides again and keeps going through the cell cycle over and over and over again. Examples of this would be like the skin cells, which are a type of epithelial cell. Your blood cells, because they're constantly getting hit as they're flying through your body, those are also going to have to be replaced constantly. And then stem cells also are going to be dividing all the time. Stem cells are your undifferentiated cells that give rise to all the specialized cells of your body. A cell then can also follow a different fate. So if it follows the fate that's on the right side of the diagram in the pinkish kind of orangey color, when a cell grows, it can differentiate, which means that it becomes specialized to do a particular function. This cell is a mature cell and it will never divide again because it is so highly specialized that it cannot make any more of something that looks like itself. So these cells that are differentiated and never to divide again, they go into what we call G0 phase, which is a resting phase, where cells leave the cell cycle. Examples of these would be like the brain and the nerve cells plus your muscle cells. There's all kinds of other different cells in your body that are too specialized to ever divide again. Some cells, though, like a liver cell, can actually be pulled out of G0 phase and be put back into the cell cycle so that it can regenerate itself. So again, basically the only cells in the body that can be pulled out of G0 phase are your liver cells. That's why your liver is basically the only organ that can regenerate itself. The first stage of the cell cycle is called interphase. Like I said before, it's made up of three parts, G1, S, and G2. During G1 phase, the cell grows, it's metabolically active, so it's doing normal cell functions like cell respiration, making proteins, etc. Once the cell decides that it's going to divide, then it moves into what's called S phase. S stands for synthesis, for DNA replication. This is where the chromosomes are going to be copied so that the daughter cells that result from the process of mitosis have the same genetic material as the original parent cell. The last phase of interphase is G2, which is when the cell prepares to divide. It's going to produce proteins and organelles and everything else that it needs to divide. And the cell is going to continue to grow a little bit so that the new daughter cells are actually going to be big enough. After interphase is over, the cell is going to go into mitosis, which is the second stage of the cell cycle. This is when the nucleus actually divides. So the chromosomes are divided up into daughter nuclei that will become part of two new individual cells once the process is finished. The process of mitosis is going to create two genetically identical daughter cells because the chromosomes that are separated into the new cells are exact copies of one another. As you get older and you get into more complex biology classes, you'll learn that there's actually 5 recognized phases of mitosis. The first being prophase, the second's prometaphase, and then it goes to metaphase, anaphase, and finally telophase, the last stage of phase of mitosis. Before we can talk about what occurs in all of these different phases of mitosis, let's talk about what the cell actually looks like before it enters mitosis. This cell you see here is actually an interphase. The nucleus is still well defined, so it's still there, and the DNA is actually going to be loosely packed in what we call chromatin fibers. So it's like this very stringy DNA. As you can see, it looks like purple strings on this cell. Once the cell moves into mitosis, prophase, the chromatin, that stringy DNA, is going to condense into what we see as visible chromosomes. So those are those X-shaped chromosomes you're probably more familiar with seeing. Then the centrioles are going to start to move towards opposite poles during the prophase stage. The centrioles are the organelles that are going to organize the spindle fibers, also known as the mitotic spindle, also known as microtubules. And these spindle fibers are going to start to form and extend from the centrioles. Also a part of prophase, the nuclear membrane is going to start to break down. This is what we refer to as a chromosome. As I said before, the chromosome X shape that you're normally familiar with seeing is actually a duplicated chromosome. The duplicated chromosome is held together by the centromere. There is cohesin proteins that are present at the centromere to hold each of the sister chromatids together. The sister chromatids are identical pieces of DNA, so it's just duplicated pieces of one piece of DNA. The centromere, again, is held together by cohesin proteins. Cohesin is spelled C-O-H-E-S-I-N. Cohesin proteins, which keep those two sister chromatids attached so that the cell can remain organized. After prophase, we move into prometaphase, and this is when the nuclear envelope is really going to start to disappear. The spindle fibers are going to really be extended and formed at this point. And the spindle fibers are going to start attaching to the kinetochore. The kinetochore is actually found at the centromere of a duplicated chromosome. And you can see in the picture with the pink chromosome at the bottom of the screen here, those little light blue proteins are actually called kinetochores. And each sister chromatid is going to have a kinetochore, where what we call this kinetochore spindle fibers or kinetochore microtubules are going to attach to the chromatid to make sure that the chromatids separate properly during the part of anaphase. So as prometaphase continues, once it's finished, you'll see that the nuclear envelope is going to all be gone, and most of the chromatids are attached to the kinetochore microtubules at this point. Then the cell is going to move into metaphase, which is when the chromatids are lined up along the metaphase plate. The metaphase plate is just an imaginary line where it's the center of the cell where all the chromosomes are going to go to organize themselves. This happens so that the chromosomes can properly separate themselves, and the cell can more easily recognize that all the genetic material has been separated. During anaphase, the sister chromatids are going to separate at the centromere. An enzyme called separase, it's spelled S-E-P-A-R-A-S-E, separase enzymes are going to come in and they're going to dissolve and break down the cohesin proteins that were holding the centromeres together. Once the cohesin proteins are gone, the centromere is going to break down and the sister chromatids can separate. The kinetochore microtubules are going to shorten in length, pulling the chromatids, now newly formed chromosomes, apart towards each pole of the cell. There's some other microtubules or spindle fibers as well called polar microtubules, also known as non-kinetochore microtubules. They're actually going to lengthen to help push the cell even further apart so that the poles can become completely separated from one another. In more detail, this is what the separation of chromatids during anaphase looks like. Currently at the start of anaphase, you're going to have a fully put together duplicated chromosome made up of two sister chromatids. But as the cohesin proteins are broken down by separase enzymes and the centromere dissolves, those chromatids are going to split from one another. We now refer to these chromatids as daughter chromosomes. They are their own individual chromosomes at this point. They are no longer referred to as sister chromatids because they're not held together. They are referred to as individual chromosomes. All right, now during telophase, the chromosomes have arrived at the opposite ends of the cell. New nuclei are going to start to reform around that DNA. The chromosomes are going to start to relax into chromatin, which is the stringy DNA, and the spindle fibers are going to start to break down because they're no longer needed. The last stage of the cell cycle, that's known as cytokinesis, is also going to start during mitosis as well, but note, cytokinesis and mitosis are two separate processes. Mitosis is the division of the nucleus, so when the chromosomes actually separate, And cytokinesis is the division of the cytoplasm when the cell actually separates. Cytokinesis happens differently in plant and animal cells. In animal cells, a cleavage furrow is going to be formed to separate the two new cells. Actin filaments, which are protein filaments, are going to form what is called a contractile ring between the two new daughter nuclei, and it's going to pinch the cell membrane together, forming that cleavage furrow. And once the cell membrane is pinched all the way and two new cells are formed, cytokinesis is complete. Now, in plants, it's going to be a little bit different because there is a cell wall, so you can't just pinch off the cell wall to separate the two new cells that are formed. So what happens is that vesicles containing cell wall material, as you can see in the bottom right picture of the screen here, the vesicles containing polysaccharides like cellulose, which is the main polysaccharide that is in a cell wall, those vesicles are going to fuse, bringing together the pieces of the cell wall. And then a cell plate is going to form that extends to either pole of the cell, creating the new cell wall that's laid down in between the two new daughter cells.